Okay, so it's a pleasure to introduce Thomas Wichmann today. Tom is the director of the Udall Center at Emory and professor of neurology there. Um, Tom originally is German, as I think most people on this call know, trained originally in Germany, then also in the United States, and has really been a leading figure in electrophysiology of the basal ganglia, particularly well known for his work on primate models of Parkinson's disease, and has led a very productive research program at Emory related to Parkinson's disease for quite a few years at this point. Also has been, I think, in a more applied way, a significant figure in trying to in both applying and trying to understand the mechanisms of deep brain stimulation treatments for Parkinson's and related disorders. So, Thomas, thanks very much for taking the time to talk today.
Right. Yeah, thanks so much for having me. So let me just see if I can get my presentation going here. All right, can you see it actually?
Not now.
Not now. Okay. See.
Okay.
This worked so well when we just
We
tried.
it worked before, right? All right.
Let's see.
No. Here we go. Okay.
So that's still the wrong mode, guess,
We've
right?
got screen sharing, but it's just a dark screen right now. There we go.
Is this right now?
The previous one was now in your presenter mode right now, I think.
Okay, the previous one was even better, huh? How about
Yeah, the
that?
previous one was, now you're on the regular presentation.
Okay, good. Let's then keep going with this. So yeah, thanks a lot for having me. Also for the nice introduction. Uh, this is about neuroplasticity and dopamine depletion models of Parkinsonism. Um, before going anywhere else, I wanted to thank people who have actually contributed some data to what I'm going to tell you today. People in my lab, Adriana Galvan foremost, she's done a lot of work in this field. Anne Carolyn Martel was a postdoc in our lab, has done work, and I will show at least a little bit of her data. Then in Jolan Smith's lab, Jolan himself, of course, and Rosa Villalba have worked in this field, and we have very good collaborators also, Hongyang Xu from Georgetown University and Bill Litton from SUNY Downstate. Many of these people are seen here in this image. So Yolan is, of course, the guy here with the glasses. This is Hongyang with another guy cool guy with glasses. And this is Bill Litton in the middle there. Funding for this work came from the NIH, specifically the Udall Center, and also this ASAP research network in collaboration with the Michael J. Fox Foundation. So, all right, so that's the thank you slides. I wanted to start with a very brief introduction. This is extremely basic, as you will see, but it will get a little bit more concrete later. And then we'll talk about basal ganglia and cortical activity changes in Parkinsonism. This is sort of this lead into the main topic, which is the plasticity in the basal ganglia, talking about the striatal plasticity, subthalamic nucleus plasticity, and to some extent cortical plasticity, and then we'll summarize this. So basic introduction is, as always, the circuit diagram of the basal ganglia and also that of the cerebellum here. So what this slide really shows is the separate motor circuitry that we're looking at. One half of this is the basal ganglia thalamo-cortical motor circuitry, and the other half is the cortico-cerebello-thalamo-cortical circuitry. They operate for the most part in parallel. There are some interactions that are now well accepted, in fact, going interactions between the basal ganglia and the cerebellum, and also between the cerebellum back through the striatum through the thalamus, actually, to the striatum. So there are these interactive pathways which may play a role in Parkinson's disease, specifically in terms of tremor and to some degree also gait disorders, perhaps. But for the most part, we'll stick with the basal ganglia thalamocortical circuitry. I will not go into a lot of the details here. It's a very, very intricate set of pathways, of course. What we're using here at the Primate Center is this MPTP model of primate Parkinsonism. It's a really well-established model of dopamine depletion in monkeys. And it replicates the pattern of dopamine loss in the human disease. So this is shown here with the DAT and TH staining that you can see. So dopamine transporter and tyrosine hydroxylase staining. In the control states, you find really strong expression of both in the striatum here. In the MPTP treated state, you find really dropout of these markers, sparing to some extent the ventral striatum, just like it would be spared in human Parkinson's disease. This is the same, a little bit at a different level further posterior, showing the putamen here, specifically in the normal and the Parkinsonian state. So it's really a blowout lesion of the dopamine system in these animals. We can monitor this in vivo using FECNT PET, looking at the DAT scan-like images, which also show dramatic loss of dopamine innervation of the striatum in these animals. So it's very good in terms of replicating the dopamine loss. It's phenotypically quite convincing. Also, these animals look Parkinsonian. They don't just have the anatomical abnormalities, but they really are very slow. They have dexterity problems. They have problems with gait maintenance and other things, freezing episodes. So that from that perspective, it's really a good model. There are many administration protocols that can be used. The one that we use is a chronic administration protocol where we inject small amounts of MPTP every week and induce this slowly progressive course of the Parkinsonism. This is not a model of Parkinson's disease, of course, so would be wrong to sell it as such. So it is a model of dopamine depletion in these animals. It's fairly specific for the dopamine system with a few exceptions. So those exceptions that are known are that the locus coeruleus neurons also are affected by this, and also that the caudal intralaminar nuclei of the thalamus are degenerating to some extent. Both also happen in Parkinson's disease, so we're not sorry about these off-target effects, but they're not dopaminergic necessarily. But for the most part, this is really a dopamine depletion model. So the firing pattern and rate changes that we see in Parkinsonism are just exemplified here. This is from the three main basal ganglia, non-striatal basal ganglia structures, external pallidum, subthalamic nucleus, and internal pallidum, that show really substantial changes in their single cell firing patterns. If you go from the normal to the Parkinsonian state, Parkinsonian state is characterized by fairly low GPE rates of firing, fairly high rates of internal pallidal firing, and also increased rates of subthalamic nucleus firing. One other thing that obviously happens here is that the patterning of spikes is substantially changed. So one big change that happens in all of these structures is that they fire more in bursts. So in the Parkinsonian state, it's well known that both pallidal segments and the subthalamic nucleus make these extra bursts, often twice the rate that you would see them in the normal state. And also that the structure of these bursts very substantially changes, actually, so that what you see here are interspike intervals. So that's along the y-axis, and this is the ISI number counted from the moment the cell was index to start a burst. So time 0 is the last interspike interval before the burst, and time 1 would be the first interspike interval within the burst. So bursts are short ISIs, of course. And one thing you can see here easily is that these bursts are often preceded by a long interspike interval, which is likely because many of them are T-type calcium channel driven. But there's also something happening before the burst, right? So there's this gradual decline of the interspike intervals, which then just before the burst, the neuron makes a longer ISI, and then it makes the burst. So there's this context around a burst that is worth looking at. And this is changing in the Parkinsonian state. So this is easiest to see here in the subthalamic nucleus. So the gray symbols are the Parkinsonian state. So it's a much greater tendency to have these long ISIs prior to the burst moment. So those things are happening. In addition to these burst discharges, we find oscillatory firing rates. So this was first at least analyzed in this paper that Guy Bergman and I wrote a couple of many years ago now. Almost 31 years ago, where we looked simply at the proportion of neurons in the subthalamic nucleus that were engaged in oscillatory fluctuations of their firing rates. And what was obvious is that low frequency oscillations are much more common than in the normal state. Again, normal is black here. The MPTP state is in gray, while the higher frequencies are a little bit less expressed than in the normal state. Similar oscillations exist in people also. And another thing that involves these oscillations is that neighboring neurons in the basal ganglia are more coupled to one another. So in the normal state, as Dieter, who is also on the call here, I think, will agree, under normal conditions, these neurons in the basal ganglia are largely uncoupled from one another. They fire in their own way. They're not really talking much to one another. In the Parkinsonian state, this really changes a lot. Neighboring neurons that you record with 2 electrodes a certain distance away from one another are often firing in these very synchronized firing patterns, which happen to be often in this low frequency range up to maybe 20 hertz or so. So you would have beta oscillatory activities here. Interestingly, in this study, they also recorded EMG at the same time and found that the EMG was also oscillatory in this patient who had tremor, but the oscillations were really occurring at a completely different rate as the basal ganglia oscillations were occurring at. So these 2 oscillatory patterns coexist, but they don't really have a lot to do with one another. So these oscillations, synchronized oscillations, have been found in subthalamic nucleus, pallidum, striatum, motor cortex, other areas. So the last thing I wanted to tell you about the basal ganglia discharge patterns, and I will come to plasticity in a moment, are that we found that they engage in sequences specifically. So this is usually when spontaneous basal ganglia firing is being analyzed, people think about it as a sort of a random occurrence of spikes at a certain frequency, perhaps with some bursting interspersed, but it is not really a structured train of spikes that is usually thought about. We found that this is actually not true. So they are embedded in a firing pattern that the basal ganglia make. There are often these little sequences of spikes that occur at very precise temporal spacing. So those can occur many times during even like a minute of firing. You would find, as in this case, there would be 9 times a repetition of the exact same little triplet of spikes that occur. Triplets are most common. Sometimes you also find quadruple arrangements or even longer arrangements. I think the longest we have ever found was a 9 spike arrangement. So they can be fairly long, these sequences. And their occurrence also changes with Parkinsonism. So they're a little bit less common in the external pallidum in the Parkinsonian state, again here in gray. They become more common in the subthalamic nucleus, and they don't change much in the internal pallidum, but there are changes in the other two structures. And we found that they sort of embedded in the overall firing pattern of cells, but then of individual cells, but they show little intrinsic structure. So if you look at, in these triplets, do short eyes come before long eyes, or vice versa, that is actually not any specific. There's no specificity to this patterning within the sequence. Of course, you would find sequences also if you just took any kind of random arrangement of eyes or spikes, but we looked actually at whether the patterns that we found were more common than one would expect them to occur just statistically, and found indeed this is a biological phenomenon. This is not just a statistical finding. So this was found with shuffled sequences, of course. Last thing about this is that they are not really an independent phenomenon. These sequences are an independent phenomenon, independent of firing rates and also of bursts. So correlational analysis really, by and large, didn't really show any specific link between those parameters. So moving on to the thalamus. This is just from our paper in 9 years ago now, where we looked at just spontaneous firing changes in the thalamus. This is controversial, so some authors in the literature have found somewhat different results, but so I'm showing you our results in this case. We found that the firing rates in the ventral motor thalamus were reduced, as one would expect it from the traditional model of Parkinson's. And the bursts and decelerations were more common under these circumstances. So bursts are more common, as you would find it in the basal ganglia also, with a greater intensity of bursting also. That's what this parameter describes. Decelerations are also more common. So overall, the variability of firing in the thalamus was more pronounced under these MPTP-treated animals than they are under normal circumstances. Oscillations were also affected. We found more power in the low frequency ranges, so alpha and beta band components of these cells, while the power in the higher frequencies were much less affected, a little bit here in the gamma band, as one also finds it in the Parkinsonian animals. Overall, these changes in the thalamus are much less than they are to be found in the basal ganglia. So what is happening in the thalamus may be to some extent a reflection of basal ganglia output, but it's not quite to the same extent there. And then the cortex was, of course, also of interest. So the cortical activity changes in Parkinsonian animals are much less explored than they are in the other structures that I've shown you. But there were a couple of things unknown. A landmark study was really this one by Goldberg in 2002, where they looked at MPTP-treated monkeys again and recorded multiple neurons in healthy and Parkinsonian animals, and were really able to show that the level of synchrony between cortical neurons was greatly increased. So this was in M1 cortex. So what they found was really that a large number of cells that started to engage in these synchronous bursts. And few years later, then a Rob Turner's study was published, also looking at cortical activity changes in monkeys, expanding this with an incredibly meticulous study. In fact, they did antidromic stimulation to identify corticostriatal projecting neurons and corticospinal neurons. So they had these two different families of neurons that they could identify. They found that one interesting piece here is that they found that these types of neurons are quite different from one another. So the corticostriatal projecting neurons are very slow in this study. This is these black columns here. So the distribution of neurons is very much towards the left side of the scale here. So very slow. And when you gave the animal MPTP, there wasn't much of a change. So you would expect that corticostriatal projecting neurons actually would change a lot because they're part of the circuit of the basal ganglia thalamocortical circuitry, but they're not really much engaged in Parkinsonism. The corticospinal neurons, on the other hand, they're faster to start with. So those are these black columns again. They're much more to the right of the spectrum here. And those neurons do actually change in the Parkinsonian state. They get slower. They also show some low frequency oscillatory activity and some increased bursting as well. So it looks really as if the M1 activity is altered in advanced Parkinsonism, and that corticospinal neurons specifically are much more affected than the corticostriatal neurons. And the last piece I wanted to emphasize before actually then showing you about the plastic changes that occur was just a brief reminder of what happened in response to treatment with levodopa of patients. So the experience with Parkinsonian patients is that they are initially quite responsive to levodopa treatment. People often go through this honeymoon period where they really feel much better, sometimes to the point that they really get rid of most of the motor signs of Parkinson. So it's a very effective treatment at that point. With the progression of the disease, changes occur. So previously levodopa responsive signs become less responsive. Levodopa unresponsive motor signs come up that really don't seem to have anything to do with levodopa anymore. And then if you start using levodopa for a long period of time, you end up with some motor signs and problems that you really didn't see before. So motor fluctuations occur, levodopa-induced dyskinesias occur, and sometimes also non-motor effects, psychiatric side effects can also occur. So if you look in the brain of animals that were treated with levodopa, Parkinsonian animals that were treated with levodopa, there's a couple of things worth mentioning. So one is that even high-dose levodopa therapy often does not fully restore motor activity. So high doses, this was a study that we did a few years back, where very high doses of levodopa were actually used, up to 30 milligrams per kilogram. This is an enormous dose, even for the monkey. Those animals did get better, so motorically, so they moved more, the activity counts went up, but they did not get back to their healthy activity counts that they had prior to getting the MPTP. So it's not fully reversing motor activities. It also was found to not normalize single cell spiking in GPE, SDN, and GPI. Atsushi Nambu, Tachibana et al. Did a study on that, and they found that the firing rates and patterns did not normalize completely in response to the levodopa treatment. And then we looked at oscillatory properties of local field potential recordings, and they got somewhat better. So you can maybe look at some of these here. So at time 0 here is where the levodopa was given, and the oscillatory activities in the low frequency ranges here that were originally pretty high really came down some, but they did not reach the healthy level. So they reached a less Parkinsonian level perhaps, but didn't quite recover to the healthy state. So the levodopa really seems to change the brain, and you may end up with sort of normalized looking movements, but it doesn't quite changes the circuitry to a completely normal state again. So it's a different state they get to. So of course this levodopa, or failure of levodopa to normalize movements completely or brain activities completely, in people, this can have many different explanations. This can, of course, can be caused by the emergence of some Parkinsonian features that really are not dopaminergic anymore, so that there's degeneration in parts of the brain that are not affected by dopamine, or it could be that there are some other medications that are interfering with the levodopa effect. So there are explanations in people. In the monkeys, this is much harder to explain because these animals have more or less a pure dopamine or strongly dopamine-centric lesion. Our monkeys do not get medications outside of these specific experiments, but usually we don't treat them with levodopa. So you don't have any kind of side effects from other medications, levodopa or other drugs. And of course, they don't get Lewy bodies or other pathology in the brain. So they just have this dopamine lesion. So, and from those kind of observations, one can come to the conclusion that there must be something else happening in response to the initial dopamine lesion that really eventually leaves the animal in a different state. So plastic changes that occur maybe secondarily only related to the lack of dopamine. And that is actually what I wanted to then talk about in the main portion of this talk. And there is a lot of evidence for these secondary changes that occur in relation to dopamine loss. So there's strong evidence that this occurs in the striatum. The first hint of this came a long time ago in a publication by McNeil in 1988, where they found this in people and humans that were Parkinsonian. What was found there is that individuals who are Parkinsonian had changes in the dendritic properties, anatomic properties of dendrites in the striatum. So what they saw specifically was that dendrites were shorter of spinal projecting neurons. So dendrites were shorter, and the segmentation, the number of segments that they would form, the complexity of the dendrite was reduced in the Parkinsonian state. That is shown here. This was very quickly then also verified to occur in 6-hydroxydopamine treated rats at the time. And later, Yolan Smith and Roosevelt Alba showed this also in monkeys. So here's a nice dendrite of a spinal projecting neurons in the control state. This is in a state where the monkey is partially depleted to the point that they actually show perhaps some cognitive changes, but no motor signs, but still they have received some MPTP, and this would be the completely dopamine depleted state when they are quite Parkinsonian. And what you can see here is that there is a gradual dropout of the dendritic spines that you would see in the striatum. So it's a very striking change in these animals. This was more recently also looked at by Steve Collier's work, where they again showed in this case 6-hydroxydopamine lesioned mice, I believe. They showed again this dropout of spines, of dendritic spines in the striatum. So the cell makes fewer spines here. What they also did in this case was that the animals were treated with levodopa, and this easily induces dyskinetic movements in the animals. And what they then found that this levodopa treatment itself also leads to plasticity. And in this case, what was happening that the spines develop these mushroom spines, so they get the dendritic spines get much larger, the remaining ones, and often there are these formations of multisynaptic terminations on them. So this is yet again a different type of morphological plasticity that goes from the normal. This is different from the normal state just as much as the lesion state is different from the normal state, only in yet different ways. So levodopa treatment can clearly induce morphological plasticity in the brain. It changes the brain morphologically. Functional changes were also identified. So this is by Surma's group, where they were able to distinguish between direct and indirect pathways and how they responded differently in these different states. So one thing they showed was that the transition from the control to the Parkinsonian state would bring with it a change in the excitability of neurons in these pathways. So what is done here is that they inject a certain amount of current and measure how many spikes that would produce. So you can do then these dose response curves where the step amplitude is changed, and you again count the number of spikes in the normal state, the black curve here. You get to this sigmoid curve. In the Parkinsonian state, in the indirect pathway, that curve shifts a little bit towards the right. So you need more current to induce the same number of spikes, which means the cell is less excitable. If you then give the animal levodopa, in this case, to the point that they become dyskinetic, so high doses of levodopa, the curve shifts back to the control state, essentially. So the indirect pathway is actually relatively regulatable and remains regulatable in the Parkinsonian state, so there's no shift in its properties. This is different in the direct pathway. So here you have, again, the control state in black, the sigmoid curve here. Here's the Parkinsonian state, where the curve in this case doesn't shift towards the right, but it shifts to the left side. In this case, which means you need less current to get the same number of spikes, so it gets more excitable. This is no longer fully reversible by treatment with levodopa. So levodopa treatment does not bring it back to the control state, but it brings it back to this intermediate state, even in this brain slice preparation. So this is again, something happened there. A plastic change happened here that means that the cell no longer is capable to get to the control state in its direct pathway. This also explains perhaps why they're dyskinetic then, because they get stuck essentially in this hyper excitable range. So those were a few examples of striatal plasticity. In the subthalamic nucleus, there's also a lot happening. There's also an interesting area to look for plastic changes. Morphological changes and functional changes. So morphological changes have been described in both the pallidosubthalamic and the corticosubthalamic pathway. So the pallidosubthalamic pathway is this main input here that provides GABAergic terminals to the subthalamic nucleus. The corticosubthalamic pathway is, of course, one of the main one of the main ways glutamate inputs get to the subthalamic nucleus, the other being primarily from the pedunculopontine nucleus. So focusing first on that GABAergic input, there were studies by Bevan's group where they looked at plastic changes there. So what they described were the way this was looked at was through among other things, through morphological methods where they used VGAT staining of GABAergic inputs. This is the red terminals here. And then bassoon staining to label presynaptic terminals. And you can see that they were green or the overlap would be yellow. So under control conditions, those terminals each make their own single one synapse. In the 6-hydroxydopamine treated state, the terminals are there, but they make now multiple synapses. This is pathologic. This is not a normal state. This is a morphologic change that has occurred in these 6-hydroxydopamine treated animals in response to the lack of dopamine. This is also nicely shown here in the figure of electron microscopy. So you have this VGAT stained terminal here, which makes a synapse with a SDN neuron. And you can see that there is just a single synapse that this terminal makes. In the 6-hydroxydopamine treated state, again, there's a VGAT stained terminal here making synapses, and this forms these multisynaptic structures in that case. So this is, again, a non-dopaminergic change that is induced by the lack of dopamine. It's very hard to study in monkeys, so we have started to look at this, at least to some extent, in our animals. One of those efforts was to look at EM changes in the subthalamic nucleus. So looking at the characteristics of GABAergic terminals in the subthalamic nucleus in MPTP-treated monkeys. So some changes were in fact found. So terminal sizes are reduced in the MPTP-treated state. Terminals are smaller, GABAergic terminals in the SDN, and the size of synapses, or the overall synaptic surface in the per terminal, is increased in the state. This is reminiscent of what Bevan's group had shown earlier in their work. So these changes are likely very similar to what the rodent experiments had also shown. And Carolyn Martel, so this postdoc in our lab, went ahead and electrically stimulated the external pallidum recording then in the subthalamic nucleus, and looking there for inhibitory responses. And she showed that under normal circumstances, the inhibition was detectable about 12 milliseconds or so after the stimulation of the GPE. In the Parkinsonian animals, the inhibition was detectable much earlier, which doesn't mean that the latency was shorter, but that the inhibition was just far more intense. So this is an intensified response, which is then easier to pick up on analysis. The duration of the inhibition was about the same in these animals. This is still ongoing, so we're hoping to publish this sometime soon. So the other area that's interesting is the glutamatergic input. So there is a lot happening there too. This is work that Abram Matai did in both Yolan and my lab. What he did was to look at glutamatergic synapses in the subthalamic nucleus. It was sort of the first look here. And he found that asymmetric synapses, which are glutamatergic in electron microscopy studies, are just the number is smaller, the density is less. So per square millimeter of tissue that they looked at, there was just fewer of those asymmetric synapses found. Those synapses can arise from either cortico-subthalamic or subcortico-subthalamic sources. So VGLUT1 positive terminals would come from cortex. VGLUT2 positive terminals would by and large come from subcortical sources, most of them probably from the PPN, as I pointed out. And what they found was really quite striking. So the half of the cortical inputs terminals in the subthalamic nucleus are lost in the Parkinsonian state, both in the sensory motor and in the associative and limbic territory. So there's a big drop-off of the glutamatergic inputs. And this is true both for the cortical and for the PPN or VGLUT2 positive terminals here, but the cortex was particularly striking. Subsequent studies in the lab had also shown that this is hard to see here, but that the corticosubthalamic terminal size actually gets larger in the Parkinsonian state, and that multisynaptic connections form, again, very reminiscent of the palatosubthalamic connections as well. And this is true for people too. So these are studies looking at material from patients with Parkinson's disease, comparing that to control. So this is a VGLUT1 stain from these individuals, which also showed a reduction of the VGLUT1 inputs to the subthalamic nucleus. This is much harder to interpret in people, though, because of the polypharmacy that they usually are under before treatment before death, when the sample is eventually taken. So there is it's not clear how this really relates to the monkey experiments, but it was good to see that it's not just a primate result for sure. In the meantime, it's actually been shown also in 6-hydroxydopamine-treated rats. This was also in Bevan's experiments, where the exact same finding was found. We looked at this also in terms of function. In that experiment, Abram recorded palatal activities in animals that were stimulated in the internal capsule. So if you do that, the easiest way a stimulus gets from here to here is via the subthalamic nucleus. So by doing this experiment, you can learn how the subthalamic nucleus functions just by recording from the internal pallidum. So we didn't record in the SDN itself because of the stimulation artifacts were just too large. So one has to take this detour to the GPI to get that information from the SDN. So this is how those studies look like. So you would stimulate in the internal capsule, record in the pallidum, and what you see there is this ginormous stimulation artifact, but then you see these little palatal spikes occurring, and what you also find is that they occur at a very specific latency after the stimulation. So these stimulation-induced spikes were prominent. We counted them as actual excitation events if they were within 10 milliseconds of the stimulation. And about 30% of all GPI neurons show this phenomenon under normal circumstances, and they often show these really huge increases of the incidence of spikes. In the Parkinsonian state, same experiment, actually same animals where we did this in, showed that just the number of neurons, the proportion of neurons that showed such responses was much reduced. So this is like half of the neurons as compared to the healthy state. Other parameters like the latency or the intensity of the excitatory spike that you get there was not changed, but just the proportion of cells was. So okay, and then I wanted to go to talk about cortical changes as well. This is the last little piece here. So basal ganglia outflow is mostly directed at SMA and M1, as you all know. So there have been, of course, these studies that Rob Turner had published on M1 activity changes in the Parkinsonian state. So we were interested in looking at morphological changes there. And Yolan did this study where they simply looked for VGLUT2 staining in cortex. So those would be predominantly thalamocortical inputs that they would identify with that. And if you compare the control slice here with the MPTP slice, it's quite obvious that there's a huge difference, right? So the control slice has these staining in the upper layers, and then again another layer-specific staining here in the lower layers, layer 5 and 6. This is really the lower layer staining is is missing. So this is a layer-specific dropout of the glutamatergic innervation of cortex in these animals. A general loss of glutamatergic synapses, not layer-specific, but global loss, had been described, actually not in the motor cortex, but in the dorsolateral prefrontal cortex. So this was a published paper by Ellsworth in 2012. Yolanda also and Rosa Villalba, they looked at the dendrite morphology. So there's the pyramidal cells make these very nice large dendrites that you can follow through the cortex, and they usually are packed with dendritic spines, as you can see here under control conditions. And just as in the striatum, these dendritic spines really disappear in the Parkinsonian state. So this becomes really depleted of dendritic spines. There's again a half of the number of spines is remaining in the Parkinsonian state. And this is a phenomenon that is found in M1 and SMA, but not in other areas of cortex. So cingulate cortex doesn't show this, sensory cortex doesn't show this. So this is a very area-specific change in the dendritic spine density. Again, it's important to realize that these are areas of the brain that are not strongly affected by the dopamine loss in Parkinson's, or in this model, in the MPTP model. There is some cortical dopamine, but the majority of these findings are very likely just a secondary plastic change that occur in these neurons in response to losing the dopamine in the striatum. So it's a circuit phenomenon, not a phenomenon that is really driven by direct cortical changes. Yeah, we're looking at this, among other things, currently by using calcium imaging techniques in monkeys. We just published actually a paper on just the technique itself and some early findings in this. So we have collected a fairly large amount of data now also on cortical changes as the animal develops MPTP-induced Parkinsonism, trying to see what sort of functional impact these morphological changes may have. Last piece I wanted to tell you about are results from mitopark mice. So this comes from studies that our collaborator Hongyang Xu in Georgetown is doing. So mitopark mice are a very well-established model now of slowly progressive Parkinsonism. They have a dopamine cell-specific inactivation of mitochondrial transcription factor A, so TFAM, that is expressed. This loss of TFAM is expressed specifically in dopamine transporter cells, from dead cream mice, which results in a selective respiratory chain deficit in dopamine neurons, so they die gradually. This is shown here. This is under control conditions. This is the mitopark mouse at 25 weeks, so there's a clear loss, and this is a gradual phenomenon, so ideally suited for looking at secondary changes after while you're losing your dopamine in the Parkinsonian state. This is a very dopamine-specific effect that's happening. These animals do really change their locomotion as time goes on, so it goes from 8 weeks down to 24 weeks, so it's a gradual drop-off of the locomotor activity of these animals, along with the gradual lack of these axon terminals. So Hongyang looked in these animals at cortical excitability. That was one of the things. So this was, again, trying to differentiate between corticostriatal and corticospinal neurons. So I had already shown you that these differ in their response to MPTP in monkeys. So in the rodents, this was then Hongyang's work. that they also showed these differences. So they showed that the excitability of corticostriatal neurons is essentially unchanged, while that of corticospinal neurons is drastically reduced. So those neurons become much less excitable. Again, likely not as a direct effect of dopamine loss in the cortex, but as an effect of dopamine loss subcortically. And this is a gradually developing phenomenon. That phenomenon alone is, according to modeling studies by Bill Litton's work, who's also part of our ASAP grant, that excitability change alone is sufficient to produce local field potential oscillatory activity in the beta range at cortical level. So it is possible that some of these oscillatory changes really hang together, are produced by the cortical change in excitability. So Hong-Yang also went on to show that these changes that occur at the cortical level may affect thalamic input to cortex. This is also an interesting and, again, not directly dopamine triggered phenomenon, where he transfected thalamic inputs to cortex with in an optogenetic experiment. So he could activate these inputs from the thalamus to the cortex optically and then record the response of cortical neurons, the electrical response of cortical neurons. So if you do this with the thalamic inputs to PT neurons, so pyramidal tracts, corticospinal neurons, these neurons under normal circumstances with different stimulation intensities, so optical stimulation intensities show different responses, so EPSPs that you can see here graded. In the dopamine depleted, 6-hydroxydopamine, in this case, dopamine depleted state, you see a substantial reduction of these responses. So there is a plastic change that moves these terminals actually, or the terminal responses actually to this much less sensitive mode, and this can be quantified. If you look at thalamic input to corticostriatal neurons, so the IT neurons, there's no difference between those control and 6-hydroxydopamine treated states. So very similar curves emerge there. And the same is true for neurons from other cortical regions that project to the pyramidal tract. So this is, for instance, the sensory cortical inputs to pyramidal tract neurons. They also don't change. So this seems to be a very specific change that occurs in the excitability and in the efficiency of synapses from thalamic input onto corticospinal projecting neurons. All right, so complicated topic, but the summary of this is that dopamine depletion can lead to secondary morphological and functional changes, and I've shown you some of the morphological, particularly because they're sort of so much in your face there, in basal ganglia, thalamus, and cortex. It's very likely that these adaptive changes coexist with changes that directly arise from dopamine loss, specifically, of course, in the striatum and in the basal ganglia areas that receive dopaminergic inputs directly. In all likelihood, these are homeostatic adaptations in the beginning. So the system addresses dopamine loss by changing its morphology and changing its function in order to compensate. And it's very, very good at that. So you can lose a lot of dopamine before you really have behavioral changes. But later on, this becomes maladaptive in the sense that it's really not, correctable anymore with dopamine replacement. And of course, dopamine replacement therapy itself can add to plasticity. So I've shown you this example from Steve Collier's work. And we all know this when you're treating Parkinsonian patients, that's just the response to the levodopa itself can change quite dramatically. This is not just isolated for dopamine, though. I think it is quite obvious to me that the deep brain stimulation will have also similar plastic consequences. It has short-term and long-term plasticity. So this was an experiment that we did some years back where we stimulated in the subthalamic nucleus and recorded palatal cells. And you can easily see short-term plasticity here where the cell initially respond very vigorously to subthalamic nucleus stimulation. So this is a palatal cell firing much faster when you stimulate the subthalamic nucleus, but that then very quickly drops off to some plateau later on, and this is reproducible. So the cells adapt in this way, and I have no doubt that this would also occur on the long term. This would be an important area of study, actually, to look at neuroplastic changes as they occur over long periods of deep brain stimulation to see what the response really does over long periods of time. So, okay, so with that, I would like to end and thank you all for listening, and certainly happy to answer any questions.
Well, Thomas, thank you very much. That was very nice. Before we open up you want to shut down your presentation so we can see everybody and people can raise their hands for comments.
All right.
So a quick question. So I guess, is your impression, from what your description, which is a really nice, in addition to your own work, a really nice overview, it really looks as if the rodent work actually is strongly predictive of what you're seeing in primates. Is that a fair comment?
Yeah, that is a fair comment. I think I mean there are clearly differences, specifically in the cortical organization that you will have to take into account, and that the territorial differences that you see in cortex sort of pervade the entire subcortical circuitry as well. But the principles are the same between the two species. It's clear. Yeah, I think so.
Okay, excellent. Yeah, that would be helpful. And I guess I'll just make the editorial comment that the dopamine signaling field is just dominated by phasic signaling. And, as you've shown very, very nicely, there are these long-term effects, which are not only poorly understood, but just really not incorporated in most people's thinking about this topic. So.
Yeah, it would also be, as I said at the end, I would be very interested in hearing more about the DBS effects, actually. So it cannot be that we apply 130 hertz simulation day in, day out for years and don't induce any changes, right? So that there must be changes occurring.
I think that cues Jerry's entrance.
Yeah, that got to get Jerry going. Yeah.
It is intriguing though, because you have all these cases where people have been stimulated for a couple of years and then the battery goes dead, like in these dystonia cases, right? And they don't change for quite some time. So yeah, obviously we're doing something. I mean, Lozano used to talk about this quite a bit, even essential tremor, dystonia, and then this work with Peter Tass, right? The coordinated reset, different patterns of stimulation producing this kind of like this prolonged benefit when you turn off stim. So you'd be curious to see what is going on there. If you could capture that, Thomas, and tell us what to do, that'd be great.
Yeah, it would be. I mean, even the morphological changes would be interesting to look at.
Yeah.
Sort of just stimulating for a month and looking before and after. So
yeah,
it'd be good to do. You had also a study, I think, that you looked at different symptoms and how that would reverse when you switch off the stimulation. So this is not just DBS, but it's also actually symptom specific that these rates of changes occur. So
Yeah, Helen Bronte Stewart had looked at that too with
Mhm.
the gait particularly, I think. And
Mhm.
she was pointing out that sometimes the gait changes took a couple days to see the full
Mhm.
effect.
Mhm.
Well, of course, this also occurs with pharmacologic treatment because a large, particularly for early PD, a large component of the treatment effect is that long duration response, which again, that hasn't, whether or not that has differential effects on different features of Parkinson's, has not really ever been examined because it's hard to study. So,
Yeah, but it's an important phenomenon, right? It can really
oh yeah.
last for many days actually before the
Potentially weeks, actually, possibly in some individuals. Yeah, I don't, again, again, there are some potential mouse models for this too. So, or mouse and rodent models for this. So this would be something that would definitely be worth studying. So,
The other aspect of this that's interesting is doing just the opposite. That's the habituation story,
mhm,
essential tremor with prolonged stim.
Mhm.
You start to see just the opposite happening.
interesting. Yeah. Okay. Any other question for Thomas or any comments?
Very nice, Thomas.
Yeah.
Thank you.
Okay. Okay. Well, again, thank you so much for taking the time to talk. That was a really fun discussion, actually. So,
Thanks a lot.
all right.
Thank you.
Thanks.
Bye.